Opportunities. Discover how food waste is not just a problem but a powerful opportunity to drive innovation, sustainability, and economic value. By Dr. Christine Bosch, Associate Professor in Nutrition, School of Food Science and Nutrition, University of Leeds, United Kingdom, on 31st of July at 7 p.m. Mark your calendars and be part of this vital conversation. Join us for an inspiring session as we dive into the world of sustainable food systems and innovative solutions to global waste challenges. International Symposium on Agriculture and Environment 2025, Pre-Symposium Webinar 9 proudly presents Finding Value in Food Waste, Transforming Losses into Opportunities. Discover how food waste is not just a problem but a powerful opportunity to drive innovation, sustainability, and economic value. By Dr. Christine Bosch, Associate Professor in Nutrition, School of Food Science and Nutrition, University of Leeds, United Kingdom, on 31st of July at 7 p.m. Mark your calendars and be part of this vital conversation.
Christine, Dr. Christine?
Yes.
How to pronounce your name? I don't want to.
Christine Bosch.
Bosch, all right. That's Dutch name. Dutch sound Dutch. Bosch.
Yes, perfect.
Dr. Sumaya, it's done.
Yeah, okay. Good evening, everyone. A very warm welcome to all of you joining us for today's pre-symposium webinar held in conjunction with the International Symposium on Agriculture and Environment, ISAE 2025. I'm Sumali Fernando, and it's my pleasure to serve as the convener for this session. We are truly grateful to have a diverse and engaging audience with us this evening. The ISAE 2025, proudly organized by the Faculty of Agriculture, University of Ruhuna, will be held on 2nd September 2025 at the Golfest Hotel, Colombo. This year's symposium centers around a particularly timely and forward-thinking theme, driving innovations in agricultural technologies for climate action and sustainability. At the moment, abstract submission for ISAE 2025 has been closed. However, if you are still interested in participating in ISAE 2025, we warmly invite you to visit our official website to find information about registration. Now, it's my honor to introduce a very special guest and today's keynote speaker, Dr. Christine Bosch, who will be presenting on the topic, Finding Value in Food Waste, Transforming Losses into Opportunities. Christine is a globally respected researcher in food science and sustainability with a focus on phytochemicals and functional plant ingredients. And also she is my PhD supervisor. So I'm personally delighted and deeply honored to welcome her to this platform. And we are equally privileged to have with us Professor Mahinda Athapattu, who will be moderating today's session. Professor Athapattu brings with him a wealth of academic and organizational experience and has been an integral part of ISAE's journey, including serving as the coordinator of ISAE 2014. And to all our participants, both local and international, thank you for being with us today. Your presence enriches this event, and we hope you find the discussion inspiring and thought-provoking. So without further delay, I now invited Professor Mahinda Athapattu to take over and guide us through the rest of the session. Over to you, sir, and thank you once again.
Thank you, Dr. Sumali. I also welcome you all to this seminar series. Next item, that is our today's session. That's on finding value in food waste, transforming losses into opportunities. Today we have very special speaker, a well-recognized speaker. Before we move into her talk, let me introduce Dr. Christine to the audience, actually our Zoom community. Dr. Christine Bosch is an associate professor in nutrition, the School of Food Science and Nutrition, University of Leeds, UK. Her research interest is mainly on health benefits in phytochemicals and other functional plant food ingredients. Currently, she is mainly focusing on valorization of phytochemicals from food processing side streams to develop effective food and feed formulation for the benefit of gut and systemic health. With that brief introduction, I invite Dr. Christine to deliver her talk today on this important topic on food waste transforming into valuable products. Dr. Christine, it's over to you.
Thank you so much for the kind introduction. 1 second, I'm just trying to share my screen. Yeah, I'm wondering whether you can confirm
Yes,
that you can see the
very well,
screen
we can see.
in full. Yes.
Yes, now okay.
Yeah. Perhaps removing the side. Okay, how's that?
Now okay, Dr. Christine, please.
Okay, so thank you so much for again for the kind introduction. It is my great pleasure to speak to you today and contribute in this pre-session to the sort of series and subsequent event. Okay, indeed, as it was emphasized, much focus at this time in my research is about valorization. Yeah, and I'm very happy to talk about this topic to you today. First of all, I would like to emphasize, as was mentioned, my research interest is in food bioactives, many of which are phytochemicals. However, not all of them. So I have a wide interest in different food bioactives in the context of health. Health does, of course, not only relate to human health, it also covers animal and perhaps others. Just to give you some overview, I sneaked in this slide from previous and current research, which we do in my group on anthocyanins, for example, on betalains, which I will refer to a lot in this talk, but also bioactive peptides, so protein fragments, they also have bioactivity. So food components that exert bioactivity, this is what interests me. And ultimately, food waste is a source of bioactives, which we, and I hope to convince you today, although we have many approaches, we still not explore to the full extent. There is a lot of excess production, side streams, byproducts, etc. They have food or feed grade. So I'm looking at characterization and development of downstream applications. Okay, to move to the topic, I prepared sort of a brief outline of the talk. I want to dive a bit into the context, food waste as a global challenge, and the need to valorize, to consider this sort of resource from my perspective, targeting phytochemicals. And I want to give you thereby some examples from recent and current research before concluding. Okay. Good. So just at the outset, we're all aware that agri-food waste is a global challenge, and it's not limited to certain countries. It has a very global dimension. We estimate, and numbers deviate, of course, depending on the source a little bit, that a third of the annual production, food production, is lost or wasted every year. And if you think of the food supply chain, there is actually production on the farm, which often is not included in the figures, but we also farm and then post-farm gate, the processing and manufacturing and retailing and consumer consumption and or wastage at a household level. This is really interesting. If we look at this figure, we can see different sort of global locations, for example, North America, Europe, and different industrialized, or what is referred to then perhaps less industrialized other regions on the globe, sort of in colors indicated what consumer, what loss and waste perhaps may be related to consumer, but in brown, from production to retailing. So it's really important to understand, that's what I'm trying to say, that the dimension of the food wastage, but also, you know, in terms of where it arises, really differs depending on where we are. There are on the globe, and there are different reasons for that. They could relate to logistics, for example, if a lot is being lost in the earlier part of the supply chain, and in the later part, okay, it's maybe more due to consumers. So that's really interesting. If you think of avoiding waste, food wastage, indeed, I had a look at the latest UN Food Waste Index report. It's important perhaps for us to also differentiate between sort of terminology. Food waste is being used to cover everything along the supply, production and supply chain, but it's really perhaps better to refer to food loss, sort of with a focus on supply, sort of like the first part, and food waste, what is really wasted retail and consumer level. So I find that very actually useful to refer to food loss and food waste. So terminology in the food waste scene seems to be changing a bit, which is good. If we look at sort of various global food wastages, the major group is fruit and vegetables. And of course, the generation of food waste, I know I defined a bit, but for ease, I use food waste in the following to refer to different aspects of it. It depends on a range of factors. Of course, the types of food and vegetable is a category, but it's very diverse, as is any other of these also. Processing stages and where we look in the supply chain. But fruit and vegetables have a huge contribution, followed by cereals and actually some other vegetables and others. If we zoom in perhaps into the fruit and vegetable category, we can again see this diversity. And we're looking at different produce of different shape, but they have sort of something in common, which is high water content and a high concentration of sort of organic substances, which is easily biodegradable if you think carbohydrates, but also lipids and others. So this is a challenge. That's a real challenge if you think of where produce arises and how it's being processed. Further, of course, there are management systems in place to manage this, but still to this date, a lot actually ends up in landfill, which is one destination, but also incineration because of the issues that come with individual wastage. For example, citrus waste is because it's so acidic, it's quite aggressive. So large part of that has to be incinerated and not landfill, because it would cause problems to the water, groundwater, but also to the soil. So different issues and different solutions. If we look at food waste management practices, of course, this is nothing new. There are the conventional applications. If you see animal feeding, this is a very traditional route, which I will be picking up on in the further. There is the landfill that was mentioned, but there is also composting. Of course, we know things are biodegradable if they are composted in the right environment. This is in the right way. This is a great solution also. And of course, for incineration, biochar production, etc. These are thermal treatment options. All of these are, of course, actively looked at for optimization, but from a valorization perspective, at different points, considering extractions or different ways of converting or redefining materials to develop a range of different products in food, in feed, in non-food feed applications such as cosmetics or pharmaceuticals. So there are lots of possibilities and a lot of work being done around the globe by different groups, of course. From my perspective, plant food waste is a huge resource, a huge dimension, underutilized. We do, as I just said, we do lots of good research and develop applications already, but it's still a lot underutilized and a great opportunity also to engage with industries to develop downstream applications. And many bioactives, the sort of interesting point with polyphenols and antioxidants in particular, which is one of my target group in my research, is that they demonstrate a range of properties. Antioxidant, which is a very generic term, but then anti-inflammatory, antimicrobial, and many other properties. And this is really interesting to consider if one develops applications derived from these. So lots of opportunities, as I also just mentioned, for polyphenols, but also if you think of bulk proteins, dietary fiber, and applications in the different sectors. Okay. I want to present an example. Sort of this is research we have been doing in my group a couple of years ago, where we looked at beetroot waste. And beetroot waste, I don't know how it applies to Sri Lanka, but in the UK, this is a huge, and in a number of European countries, beetroot crop production is increasing because of the demand on products such as beetroot juice, but also other products, which leaves behind a lot of waste, for example, the juicing industry. But we also face, in fact, a lot of overproduction. This is this sort of pre-farm gate wastage, overproduction, oversized beet. If they are too big, what particular retailers want, they get rejected and then be, in many cases, thrown away. So at least in the UK and Europe, this is a real problem. So we did quite some extensive work on understanding how we can utilize this material, extraction work, and stabilization work. For example, you can see on the pictures that the beetroot is not very stable. If you see the discoloring, depending on the temperature you store your extracted material, you are losing this sort of red color, red pigment, which is a major attraction sort of in beetroot and of interest from different purposes. So we did extensive work in understanding different sort of beetroot fractions from powder juice and differently dried wastage material, FDs air dried and then freeze dried under protected conditions. And we identified, in fact, just to show you some results which we gained through this, or knowledge which we gained, important knowledge which we gained through this research, is that the red pigments in BW is the waste beetroot material dried under protected conditions. So this is a really good source of this red pigment if you think of coloring industry, but also other purposes for extracting that. So there is no reason why pulp waste could not be used, but it also still contains a lot of other polyphenols and has important antioxidant activities. So we think this is a really good material. So this was sort of one of our early findings, and we really explored that in different directions. And this brings me to the application sort of in animal production. There's a huge interest in the sector, in the animal production sector, in utilizing materials from waste of fruit and veg, but also others, to improve meat, to improve the animal, but also to improve the product we get, the meat, for example, storage, shelf life, etc., all of that. So there's a huge interest in the health of animal and the productivity of animals. And if you look on the right side, just the below image, traditionally, this is nothing new. You would think, yeah, traditionally this was done, scraps and excess food was fed to pigs or other species, but in a highly controlled, in an environment where we need to increase, think we need to feed an increasing global population and a very controlled, hygienic environment. This sort of applications are not possible. So you need to identify other ways of redefining materials in order to integrate them into, for example, feed in this highly controlled environment which we are facing in most countries globally. I'm moving sort of with this slide to some of the piloting research we've done some time back, also a few years ago, where we explored really the application of beet. Just demonstrated you sort of where we think there is a lot of value in this material, in the application into animal feed, and in this case, pig industry. I don't know about Sri Lanka, but a pig industry in the UK and in many European countries is a very important animal, and a very important sector in animal production, next to poultry and some others. So we were really interested to explore sort of the health benefits of adding bioactives from beet into the feed of animals. And we were focusing, and this is what you can see on this slide on the left side indicated, we were focusing on a young animal, on a very young animal. We call this the weaned pig, which just is 20 at this around a month old, and leaving sort of the mother environment, being transferring into sort of solid feed in a group situation, sort of separated from the mother, different herd and different food. This is a very vulnerable period where the farmers lose the animals up to 20%. And in the past, years ago, antibiotics were often added in the feed to prevent any issues, because the issues arise from the gut. So different, as I said, solid milk to solid feed. This is a huge challenge. And then the social and other challenges, they lead to a disruption of the intestinal homeostasis, inflammation, diarrhea, mortality increase. So that's the problem in feed antibiotics. This means a certain amount added in the feed that would help, but that's not allowed. As we know, antimicrobial resistance is one of the reasons, or major reason for that. The replacements for antibiotics are zinc oxide and similar products. They are not allowed since a few years, also now because of the environmental contamination with zinc. So there is a huge interest to explore and develop solutions that can be added into the animal feed. There are safe, beneficial, but natural based. And this is what we explored with the beet, because we have identified in our research, it's very potent anti-inflammatory, very potent antioxidant, and we want to understand whether that can benefit that gut situation. So we designed a trial in these young pigs. This is 2 days, 2 weeks is a typical time, and then you monitor how these animals grow, how healthy they are, how diuretic, and then from sampling, we can look at gut inflammation, microbiome changes, and also systemic markers of health. So this was a really, really important pilot trial, and it demonstrated that actually the animals, from a health perspective, they were actually all quite healthy. So the real difference we can see when we look at gut microbiome outcomes. So that's a really good outcome that can be modulated if you add, if you change feed inclusions or bioactives in feed. We can see some indicated some data on the right side. Control is always the control, of course, CON. Zinc oxide, you can see that this effect is strongest, and RB2 and 4, these are just additions in 2 and 4% of our red beetroot preparation, which we there added. So we can see there are overall, we have a lot of other data on that, of course. Overall, we like to think that the changes in the 2% addition of beet is most similar to zinc oxide, so that's a good thing. And we can also see that, for example, when we look very specifically using like real-time PCR and similar methods, when we look at bacterial presence in the gut, that enteropathogenic bacteria are lower under these conditions. So there is potential. So this is piloting research. There's not there was not really anything on this specifically to compare, but we believe that there is potential for sort of inclusion of beet and perhaps similar formulations in animal feed. Based on these initial piloting research to understand the properties of beet and its potential in animal feed inclusion, we were fortunate to get large project funding for a project here, the SNAP project, Sustainable Nutrition for Animal Protein Productivity, which really targeted what we had piloted earlier. But we were not limited to beet. Yes, we included beetroot, but we also had citrus, we had berry waste materials from, for example, blackcurrant production, which is big in the UK, but also citrus. You may think, in the UK, it's not quite the climate to grow oranges or similar, which is true. However, we have large companies in England that are actually pressing juice for the supermarkets in the UK. So we have a lot of wastage on the citrus side. We were working in these projects on different, so this is part of the innovation in the project, on different ways to extract, to green, to sustainably extract, to stabilize. We know that bioactives are not very stable, to encapsulate perhaps. So yeah, in different ways, trying to stabilize the material, that it could be, for example, microencapsulation, or so that it could actually be incorporated in a good way in a feed formulation. If you speak to feed formulators, they have their recipes, so you need to have ideally in a solid powderized format. You need your sample that it can be incorporated in the mix, and they also want to pellet it, which is a country-specific thing. Pigs in the UK are usually fed on pellets. We were targeting with that also the young pig, because we wanted to explore further what we had established in our piloting research, but we were also looking at the end of a pig's life, going into sort of animal meat production. What is this shelf life? It's really changed, sensory properties, etc. So this is actually work that is mostly still ongoing, and we have seen that in the weaning pigs. We have confirmation that a number of supplementary bioactives we add into the feed, again, are able to change, in particular, the gut microbiome sort of composition. And in the finisher pig, whilst I can't show you detailed results, we are seeing that the addition of certain bioactives improves the quality of the meat. Sort of, if you think what are quality indicators, it's microbiology, for example, but it's also rancidity, oxidative stability, and this is really exciting for us to go forward. We did, as part of this research, a lot of, I mentioned encapsulation. We did a lot of microencapsulation, sort of where you use spray dryer, which you can see sort of schematically on the left, and we were trying to optimize some of the conditions we use to understand what the best conditions are in terms of temperature and ratio between, well, between, if you microencapsulate, you have your bioactives in an extract or a juice or a solution, and then you have your matrix, which could be protein or it could be starch-based or other. In this case, we did a lot of work with maltodextrin that worked really well, but also whey protein and some others, and you can see the outcomes on the right side, depending on the matrix. I think the top one is yeast that didn't work as well as protein and starch matrix. So very important also to optimize these conditions. And we've learned a lot while we did this research because we identified actually that the scalability, that the scale we need to do animal trials with a normal spray drying technology is also not economically very feasible, apart from not being easy to scale up in the first place. So in these projects, they were actually collaborative projects with industry. Our industry partners, they developed methodologies to combine, to generate hybrid material. If you think of microencapsulation, you're generating, let's call it hybrid materials, and it could be different sources of bioactives, it could be different matrices. So we developed a method, a workflow that allows actually to do, it's not spray drying, but it's a way of preparing the materials, generating hybrid material. Okay. Hybrid materials without the spray drying, without the high temperatures. And you can see this was actually very colorful. So we did a lot of analysis related to that. When you include it in the feed, if you look at the picture in the middle, you actually don't see a little bit of difference, but not as much difference as we of course see in the original sample. And you cannot include huge amounts. You are looking at a low percentage to include these. So very exciting. And these were incorporated into feed at different age groups also. So this is still ongoing sort of analysis of these. But excitingly, I think I indicated earlier that the incorporation we've just presented, this is one of the first data sort of we were able to show or indicate at the conference, that we can see that the different additions of the different materials into the animal feed really confirmed that we can see changes in gut microbiome profiles at different levels, depending in what detail you look into these, just to give you some snapshot on that. So we think there is a huge opportunities in particular in animal feed applications. Of course, when you do this type of research, you learn also a lot on food and feed grade and the requirements, to be sort of approved in individual countries for individual applications. But a lot of the material indeed has that is generated in food processing is food and feed grade, and in particular in animal feed, there are huge opportunities for the different species to develop tailored solutions. There may be additives or supplements. That's also a regulatory difference. But I think it's very exciting space sort of to be active in at this moment in time, because as you can see on the right side, you need to refine the material and trigger out the properties or the attributes you perhaps want, and to ensure that they are included then in the feed formulation that you are preparing. To add on, we were able to get a second project in this space, conceptually similar, but we were utilizing fermentation. We were also looking at different waste streams. and we were using fermentation as a way to stabilize initially material, but thereby you are also providing viable, well, during the fermentation, you increase the bacterial, in that case, presence, which is also something you want, given that probiotic feed supplements are very popular and very effective, but it's also the metabolites these are generating. So it's a different type of material, in that sense, enhanced, but also to be incorporated into animal feed. So we're still analyzing also data on that, but that was very promising looking from the start. And our latest project, which we got based on the same concept, on the valorization of phytochemicals from excess production, side streams, etc. But that is food grade, and to target basically to develop prototype ingredients, prototype products, where we can incorporate these bioactives that we have recovered from the waste material, but develop applications that benefit gut health. And this is a very more mechanistic sort of project, but we are targeting sort of the mechanisms that I mentioned earlier in the context of the animal applications. Is the gut inflammation, is the permeability, and it's the sort of gut microbiome and dysbiosis, which we find in humans. We refer to leaky gut. A lot we find this in the chronic disease and aging conditions, and it is something which we are targeting as part of that research with our prototypes. So I hope that kind of for us, this is really exciting to be able to develop feed, but also food from food grade material, also applications which could really lead to functional food products or supplement applications, which is open in this project, but this is really ongoing. I don't want to leave the presentation without referring to challenges if you work with this material. There are huge, of course, raw material issues, as you can imagine. The material is some of it is particularly short-lived, but they all are, with a high water content, low dry matter, so that's really easy to be contaminated, or intrinsic yeast or bacteria just grow away very quickly. So there are perhaps also other contaminants, and of course, or an issue which we also see is that there is a lack of audit trails, sort of, at the moment at least. Processing related, of course, if you think, oh, do you need to extract, or can you use the material as is, or any other manipulations? They could maybe come in with chemicals. How do you deal with that? We in our case, conceptually, we also had the ambition to be very green chemistry, sustainable with the use of that, but generally that's in the space a problem, of course. And then whatever you extract, you may also enrich the contaminants and any anti-nutrients. So there's lots of things to consider working with this type of material, but nevertheless, I think it's a huge opportunity, and that takes me to my last slide, to really summarize as much as possible that I think fruit and vegetable waste streams are highly valuable sources of phytochemicals. So we are wasting this waste to large parts. There's a lot that's going on already, but a lot of room. So the nature of the material requires quick access, localized solutions. Some of is easiest if the materials are homogeneous, but often we have heterogeneous side streams. So if it comes from primary processing, for example, then it may not be it may be a mix of different veg, etc. Many applications are there beyond feed, in food, but also non-food, cosmetic, etc., yet to be explored. Materials, of course, need to have the right quality, food feed grade or pharmaceutical grade. Safety concerns will need to be addressed. So lots of opportunities, in my view, for exploration, innovation at all stages of the process. And of course, we need circular interdisciplinary approaches to achieve a maximum output and impact in these days and times. There are lots of projects that have focused only on the one aspect, but what we need is projects that are much more circular and interdisciplinary, involve LCA, for example, life cycle analysis, to really actually quantify the environmental and sustainability angle as well, which we have done in our projects. I didn't show you that in particular. So we need to determine that as well. And that really brings me to the end. I thank you, the audience, for listening, and I'm looking forward to any questions you may have. Do you want me to stop sharing?
Maybe you leave the presentation on the screen, because maybe you can refer to the slides when questions are asked.
Okay.
Yeah.
Yeah. So I leave it. Okay.
Yeah.
Yeah. Okay.
Yeah. Thank you, Dr. Christine, for very informative and
Yeah.
insightful presentation. That's a really important on Sri Lankan context, because we are food waste. It's a serious issue. In Sri Lanka, food waste accounts for 1/3 of total food production, which is a really big issue. And also, it's very much in line with our symposium theme and the research thoughts of many of our academics in our faculty. I'm sure our Zoom community has a lot of questions. You may ask questions, raising your hand, or maybe you can just drop a question into a chat box. Yeah, Dr. Christine, to open the discussion, let me request one clarification, because your talk is very much relevant to my field as well, animal feeding. Now, in Sri Lanka these days, we are very much concerned, or we are very much interested in black soldier fly production. Now, when we extract these bioactive compounds from this food waste, now what kind of residue left behind? Can we use that residue for that kind of production, black soldier fly production, or some other uses?
Yeah. Yeah, the black soldier fly application is indeed very interesting, and is also popular in among some of my colleagues. I guess, yeah, I think there are applications with black soldier flies where they're specifically considering the bioactives to be actually, improving. It depends what you want to do with the larvae or otherwise. But in any case, if the substrate you need to feed the larvae, if that is mainly lipid carbohydrates or proteins, etc., sort of, they would, if you extract, for example, bioactives, this is typically not necessarily water, but water solvent mixtures, or so that would leave behind everything else if that were to be done. So anything that's soluble in that would come out, but the rest would be well suitable, I guess, for black soldier fly applications. Yes. Yeah. But it depends on the specific sample you look at. I don't know what you would look at as target requirements for protein or other nutrients in the black soldier fly.
Basically, we're concerned about the carbon to nitrogen ratio. We are not targeting that much of nitrogen, actually. So carbon
So you don't
nitrogen
want too much, yeah.
No,
Okay.
we don't need that much to say about
Yeah.
carbon nitrogen ratio, about 15 to 20. Yeah, according to you.
Yeah, that's also really, really relevant, I guess, because there's a lot of interest also to recover proteins. for different applications. So if your black soldier fly doesn't need as much protein, then you can see it as part of a circular approach. Yeah, you try to recover some maybe higher value compounds for food or whatever food feed cosmetic application. Then you consider the protein, which goes in different applications, and maybe remainders may actually be well suitable for black soldier fly. Yeah. Mm.
Yeah, that's what came to my mind about the residue of extracting. Yeah. Any other?
Yeah, perhaps while we wait, of course, the project sort of concepts or what I was talking about. My background is nutrition, and I've been focusing on the bioactives and the properties for many years. But it's really exciting if you work in a more wider project context, in interdisciplinary research projects where you have, because I need the chemists, I need the animal scientists and food colloid colleagues for encapsulation, sort of. That's really exciting space because you can work as teams and you can achieve so much more.
Sure, exactly. Yeah, spot on. Yeah.
Yeah. Are there any questions? I'm not sure if I can. Okay. We often see why food scientists work on developing value-added products from food and vegetable waste. Health professionals focus on increasing fruit and vegetable people. Yeah. What, in your opinion, is the core issue behind this paradox? Yeah, so I think I'm trying to work in the in-between space to exactly address, I guess, that, to combine well, what food colloid, food chemistry, food sciences are often working on. But an issue, I think, speaking also to industry in this space, if you talk about food waste or whichever way you phrase it, it seems like, oh, this is not, this is it's contaminated, something that's not good enough for consumption, etc. So you think, so I think this is probably part of the issue, that ultimately you're working then with material that is being redefined, redesigned. But in any case, concerning, of course, is excess production. And the fact that then retailers who package, for example, if there is this oversized beet, is maybe a good example. If I don't know how it's in Sri Lanka, in the UK we can buy pre-cooked beet in packages and they have a certain size. So if they're oversized, they're not just being cut, they're being rejected. So that's a very obvious sort of oversize example. So I think, but there's nothing wrong with it. There's nothing wrong with the beet. So it's understanding that, and maybe consumers being more flexible, that not everything needs to be looking or should be looking the same. And of course, the excess production on the farm, we know this is a lot linked to farmers, and this will apply everywhere. They need to plan a certain margin of excess. If there is anything with the crop and the carrots or whatever are not growing as well, they still need to deliver on the volume. So factoring in 10, 20, 30% more to grow, it's just a thing that then there is no destination for it. If the manufacturer or retailer only takes then whatever was agreed and not more, then we need to find solutions or help the crop producers, the farmers, to develop these solutions. Because there will always be the unknown, especially under this changing climate, heat, hot or heavy watering, whatever periods, to be able to deliver what they've agreed they would with the manufacturer and retailer. I hope that kind of answers that. In many developing countries, significant post-harvest losses of fruits and vegetables occur due to poor handling, storage, transportation. What low-cost sustainable strategies can be implemented? Yeah, there's a lot of emphasis on this kind of post-harvest space at the moment. And this is, if you think of some of the initial slides, this is a particular issue in some countries, and it may be indeed, as is mentioned, be due to the logistics. And that, I mean, that is maybe very country region specific things, depending on how transport ways, storage capabilities are, if that can be improved. If you think of fresh crops, if you can't deal with them fresh, what can you do? Drying is perhaps a way. Can you make locally? Are there ways of if the logistics is not easy to improve, are there ways of locally dealing with some of the things? And it could be drying, could it be fermentation? So we were really keen to work on that fermentation aspect in one of our projects in order to be able to develop local solutions. Although not an issue elsewhere, but it's also in other countries, it's about transportation cost. The volumes that need, then if they can be transported, but you would want to not transport. If you could dry or do whatever, maybe you would reduce it to less than half of the weight. That would make a huge difference to cost. So it has different dimensions, that issue. But yeah, really important. Thank you for bringing that up. Yeah. So yeah, there are lots of groups, I think, trying to look into this sort of post-harvest, reducing that loss.
There are a few more questions on chat box.
I can't see in the chat.
I do. Oh, at my side, there's a one question. Yeah, that's again comparing bioactive compound extraction against biogas production.
Mm-hmm.
So what we can is the attendee suggests that what we can do is biogas production. What's your opinion?
So what exactly is the question? Sort of,
No,
if you think.
It says
Yeah.
suggests that it implies that it says what we can process using that fruit and vegetable waste. I mean, we can use that for biogas production. I think he is suggesting biogas production over phytochemical extraction.
Yeah, I think it depends on the material, but I think it's really important. So if you think of biogas, that's maybe a final destination. So if you think of, depending on material, what could be valuable components, and is there a way of recovering them somehow? So it of course depends on the group of compounds. It depends on the value that these have or could potentially achieve. Is it in the end economically, is it technically, is it economically feasible? So I think there is a lot of room for exploration, but I think projects these days, if possible, should suggest to look at different different categories of compounds and develop a more circular, not a linear approach. How can we maybe take ideally bioactives or groups of bioactives out that have a higher value and convert them into something useful? And there may also be protein, fiber, other useful ingredients that could be used for developing biofilm or other applications, and whatever remains, can that be decomposed? Can it be going in bioenergy, biochar production? So I guess it will depend on the type of in this case fruit and vegetable material.
Yeah. So there
But
are two more
possibilities. Yeah.
Yeah, lot of possibilities. There are two more questions. One question is, what are the toxicological risks and health effects on animals fed with food waste containing contaminants or pathogens, and how do how do these toxic effects impact animal welfare and food safety? There's another question which is also related to that one. It asks, is it safe to use rotten leftovers such as fruits or vegetables, maybe to
Yeah.
extract bioactives?
Yeah. So it's really important to think about the quality of the material. So if the material is already rotten, then you've overtaken, it depends on material, but yeasts are growing really quickly on material, depending on the conditions, what direction these go. And you can assume that you have some buildup of unwanted or even toxic components. If you ideally access the waste material, and for example, we now in the UK grow grapes for wine. We have some wineries. So the grapes are pressed and the skins need to be collected within a few hours. Otherwise they would be too overgrown with yeast, etc. already. So it's really important, the logistics of the material, but also then developing, if you ferment something, for example, if you could add Lactobacillus plantarum or similar to your material, then you would actually achieve a preservation of the material relatively quickly, and then you have more time. So you need to buy yourself time often. So you can't just add in a commercial animal production, the material will have to be analyzed. So positive release, for example, if you produce it, the absence of pathogenic microorganisms, that often needs to be tested or usually needs to be tested. There will be country probably specific legislation on that. But the commercial feed formulator, formulators, they need this information, otherwise they should not be adding anything in the feed. Yeah. Does that answer, I think, the question? Yeah. So in a commercial environment, production, that will all be controlled.
Yeah.
And this material we are working on that went into animal feed, that was all tested for absence of concerning organisms. Is it safe to use rotten leftovers such as fruits or vegetables? Yes, so that kind of comes back to that. If it's already rotten, so you're working against time. So if it's already rotten, it's problematic. I would say no. So that's why, it's important to develop to think about local strategies also. If you think of animal feed, you could also think about silage. I don't know how relevant that is in Sri Lanka specifically. We were working in a project where there was some incorporation tested of waste, food vegetable waste, into a typical silage. That's also options. So there are, I think. Is it safe to use? So that was the same. I think we've answered that. Okay.
Christine, what is the bioactive or phytochemical extraction that is effective against the leaky gut condition?
Can you please repeat that?
No, what is the bioactive compound or the
Yeah.
phytochemical that extracted from food, food waste that is effective against leaky gut condition
Yeah.
in animal?
So leaky gut is defined as part of it is hyperpermeability. If you think of your gut as a barrier function, so permeability is impaired. We have inflammation in the gut and we have a dysbiosis, so the balance between good and bad bacteria is not ideal. So these together sort of are being described as leaky gut, and we observe that in aging, increasingly in with age, but also chronic disease conditions. Heat stress also causes leaky gut. So if you think of these mechanisms, inflammation, gut microbiome, but also gut barrier function, all of which there will be literature on bioactive compounds, different polyphenols, but also others being effective. Many polyphenols are anti-inflammatory. So, for example, we work a lot with berry waste now. So we have a lot of anthocyanins in the material, and we know that's very that's good anti-inflammatory, but also actually from a gut fermentation perspective, anthocyanins are wanted there as well.
Thank you. Dr. Sumali, can we permit for any more questions? It's almost 8:05 here.
Yeah, I think now we can conclude the session. I think. Yeah. Thank you very much, Professor Tapattu. So on behalf of the ISA 2025 Organizing Committee, I extend my heartfelt gratitude to Dr. Christine Bosch for sharing her valuable insight on finding value in food waste, transforming losses into opportunities. We truly appreciate your time and expertise. And also, I would like to thank Professor Mahinda Tapattu for expertly moderating today's session. Your contribution was invaluable in making this event success. And to all our participants, thank you for joining us. We hope this session was insightful and beneficial to you all. And a big thank you also to everyone who worked behind the scenes to ensure a smooth event. And before we conclude, a quick reminder, e-certificates will be issued via the Lanka Education and Research Network through the Micro Rewards platform. Once ready, you will receive an email at the address provided during the registration. And don't miss our final pre-symposium webinar scheduled for 7th August. We warmly invite you all to join for that session as well. And also we encourage you to stay connected with us through our website and social media platforms for updates and more details. Once again, thank you all, and we look forward to meet you again soon. Thank you very much. Thank you, Christine.
Thank you all. Thank you.
Thank you. Thank you so much.
Thank you.